So good morning, everyone. I am Mark Pettis. I serve as the medical director at Functional Formularies, and I also serve as director of medical education and population health for Berkshire Health Systems in Western Massachusetts. And in that capacity, do a lot of community-based consulting and develop many of our integrative medicine programs, all of which focus heavily on nutrition. And it's great to be able to share this topic with you, and thank you all for taking time out of your busy, hectic lives to tune in. I also serve as the Associate Dean of Medical Education at the University of Massachusetts Medical School, and this is one of the more hot topics, without question. And though there are still far more questions and answers with respect to the role in a more specific way that the human microbiome plays in human health, there are a lot of insights that continue to emerge rapidly each day, and I'm glad to be able to share that with you. I would say of all of the research now in the life sciences, which are exploding, none is capturing more attention than this. And this fundamental shift as we move from the microbe as a pathogen to the microbe as a commensal organism that very much is part of human biology is very much shifting the way we think about who we are as human beings and how we function. And as it turns out, these fellow travelers are very much a part of who we are, and I would make the case that we are indeed composite organisms. So I've done some work at Kripalu. This is a center for yoga and meditation not far from where I live in Western Massachusetts, and I was once asked who the most influential yogi in my life was, and at the time it was this guy here, Yogi Berra, who once said, "The future ain't what it used to be." And I love that quote in that there is so much in 2017 that I believe is revolutionizing the way we think about human health and how humans function and interact in relationship to the environments that they are in, that it's very much changing this landscape of what health is, how can we create it, how can we more effectively not just manage disease but prevent disease. And my background's in internal medicine, nephrology. I practiced nephrology for many, many years and dealt with very severe end-stage renal disease and many chronic complex medical morbidities. And it's really been over the last, I'd say, 15 years that integrative medicine, also of which I am boarded in, has just fundamentally changed the way that I help people. And I think Yogi had it right. There's so much today that one would have never predicted 5, 10 plus years ago, and one can only imagine what the future will continue to bring. So 3 fundamental objectives for our precious time together. One is that I certainly want to create some understanding with respect to what has driven what I believe a loss of legacy, a microbiotic legacy over the last couple of generations, and how is that shift in legacy, loss of diversity as an example, linking with what we're now beginning to understand to be many mechanisms that seem to be at the underpinning of health and chronic complex disease, things like inflammation, things like detoxification, things like vitamin production, vitamin K, B vitamins, etc. There are many links that are now being described, I think, with much greater clarity that makes clear this connection as a fundamental underpinning of human health. And then we'll focus on what I believe for all of you will be somewhat of a review with respect to lifestyle interventions that can restore balance and health to the gut microbiome. And again, the research is still very much evolving here. So I think this is a story that will continue to shift dramatically with every passing week and month. So if I had to summarize the greatest breakthroughs in our understanding of human health over the last 10, 15 years, I would say that it would look something like this. We know that our environment, and by environment I'm referring to the environmental inputs in our lives, how we eat, how we move, how we interpret and respond to stress, how we sleep, how socially connected we are, how much love and meaning we cultivate in our work, love, and play. All of these aspects of our environmental inputs we know are now very fundamental in changing the way that our genes express themselves. So epigenetics, a revolutionary shift in how we understand gene-environment relationships, is clearly suggesting that lifestyle and all elements of lifestyle have the capacity to change the way our genes express themselves. So in this emerging model, we are not necessarily prisoners of our DNA, or DNA does not have to become our destiny. This is a very dynamic, malleable relationship where we can take genetic predisposition and shift that expression at many levels over a relatively short period of time. And then you have the microbiome research and this 100 trillion community, largely in our colon, but obviously on our skin and our oral cavities and the perineum. And again, the research is quite clear now that we are composite organisms. This community is not just coming along for the ride. They are instead playing a fundamental role in all aspects of human biology and human health. And then all of that, I would in some way suggest falls under the category of spirituality. We all have our unique ways of describing that or expressing that, but this, in my humble objective opinion, brings us into the realm of the quantum, and it's an interesting place that does not obey the general rules of Newtonian physics. So in this way, our thoughts can influence DNA expression, can probably impact the microbiome, and things like love, compassion. These non-local experiences of life probably are having profound effect on human health at the level of vibration, at the level of light, at the level of electromagnetic frequency. And that will be a topic for another time. There are many nationwide and global projects now. This is just one, the Human Microbiome Project that David Relman at Stanford is overseeing. Many of you I know are familiar with the American Gut Project, Ubiome. These are research infrastructures that are leveraging really this new world order of citizen science, people contributing samples, getting results, and beginning to try to identify patterns. But we know today that if you look at the entire genome of the human being, of the Homo sapien, literally 99% of that is expressed at the level of the microbial world. And interacts very intricately with the human microbiome. We know that digestion, the extent to which calories are extracted from the foods that we eat, are also greatly influenced by the quality and diversification of the microbiome. I'll show you a snapshot of some data looking at obesity versus lean individuals. Detoxification and elimination of toxins very much enhanced by the quality of one's microbiome. We talked a little bit about epigenetic expression, and if you look at, for example, butyrate, which is a short-chain fatty acid produced as a metabolite interacting between the microbial community and fermentable fiber, that butyrate is much more than a health-promoting fuel for the colonic epithelium. Butyrate is a signaling molecule that will influence gene expression on an epigenetic level. we know that one's immune system, and this is probably where the research has been most predominant, but we know that the human microbiome plays a very significant role in regulating one's immune response, how tolerant one is to their environment, or on the other end of the spectrum, how ultra vigilant their immune systems might be, and in those instances might take the form of autoimmunity or celiac or gluten sensitivity. And we know that this immunomodulatory regulation is systemic, so that which emerges in the mouth, on the skin, in the gut, very much will influence, from a systems biologic perspective, inflammatory pathways throughout the body. So in that context, the human microbiome can influence insulin sensitivity or resistance, as well as leptin sensitivity or resistance. And we know these are major players in all aspects of health, of weight and metabolism, cardiovascular disease, etc. Vitamin modification, whether it be B vitamins, vitamin K, neurotransmitter function, particularly the aromatic amino acids that are so important for dopamine and serotonin, are largely influenced at the gut level. We'll look at some of the research looking at the short-chain fatty acids, the acetate, propionic acid, butyrate, and their emerging roles, again, not just as metabolic byproducts of fermentable fiber fermentation, but as signaling molecules that will influence inflammation, influence gene expression. And we know that the gut is a very active neurologic organ. System can very much communicate via the vagus nerve, and many animal models looking at the role the gut microbiome plays in hunger, metabolism, obesity are often mitigated when you sever the vagus nerve. So that's an important means of communication between that environment and the central nervous system. There are a lot of reviews, and many of you, I'm sure, are seeing reviews every day in your work. This is a more recent one that is, what I think, sheds some nice light on some of the emerging physiologic, immunologic, and metabolic processes in human health, and how alterations of the gut microbiome, and particularly the intestinal environment, from permeability to dysbiosis, is very much a driver of human health. And as you all know, we live at a time now where the burden of chronic complex disease has never been greater. And despite our health system sophistication, we've really done very little, if anything, to stem the tide of these chronic complex diseases, from obesity to diabetes and insulin resistance, cardiovascular disease, cancer, Alzheimer's, etc. These are things that I see every day in my practice, and I know you are seeing as well. And what's fascinating when you begin to really look at these relationships is that our food supply is so poorly designed to promote the health of the microbiota. I mean, who thinks about this? Most clinicians, until recently, haven't ever really thought much about the role the microbiota plays, or the relationship between the food that we consume and how it impacts that. But in a very stunning way, 75% of the food in the Western diet, the standard American diet, is of limited or no benefit to the microbiota. And I would add to that, not only is it limited or no benefit, but it's probably quite harmful. So there are clear connections. And as profoundly important as nutrition always is, this extends well beyond any discussion of vitamins or minerals or phytonutrients. This is an entirely new foray of how food interacts with an ecosystem that is largely contingent on the extent to which fermentable fiber is present. And we all know how deficient most Americans are in that respect. And if you look at the microbiome from DNA, RNA stool analysis of a more ancestral culture, and this particular paper looks at the Burkina Faso in Africa. And if you look at the upper right distribution, and I know this is a little bit confusing, but the point that I wanted to make is that the preponderance of species, and there are well over 1,000, 1,500, and the list continues to grow, identifiable species in the gut microbiome. This is a predominance of Bacteroidetes, which is a phylum, relative to the Formicides and the Burkina Faso group. And what you don't see here, but if you could, it would be much more diverse. Well over 1,000 to 2,000 different species of organisms. Now, if you compare that below to a typical Western European, which is just a European surrogate for the standard American diet, you very much see a shift in the preponderance of the Formicides species or phyla, with the Bacteroidetes. And again, not demonstrated here, you would see far less diversity of species. And not surprisingly, these are cultures that do not eat processed foods. The grains that they eat are very coarse whole grains, very different than refined flour-based foods, which are essentially stripped of nutrient density and fermentable fiber. Great deal of diverse plant-based intake. And we know that most ancestral cultures around the world, while they may have significant variability of the macronutrient proportions they eat, some eating more fat, some eating more carbohydrate, what you do see in all of these different ancestral cultures is a very high amount of fermentable fiber consumption, often well over 80 to 100 grams per day, compared to the standard American diet, where many people are getting less than 10 to 15 grams of fiber per day. And when you look at, this is the same analysis from Burkina Faso, if you look at the short-chain fatty acids, these byproducts of fermentation between the gut microbiome and the fiber in the foods that these people are eating, on the left you see in the black bars the amounts and proportions of these short-chain fatty acids. In general, across the board, compared to the gray, which is the Western European, you see dramatic differences in the amount of short-chain fatty acids produced. And again, we know that many of these fatty acids play very important signaling roles with respect to human health, inflammation, mitochondrial function, and epigenetic expression. And then on the right, and this again I think is typical from a lot of what has been published, you tend to see on standard American diets, or in this case sort of a Western European diet, disproportionately high amounts of these enteric organisms like E. coli or Klebsiella. And while these are organisms that are ordinarily present in all mammals, you see very, very high percentages in folks that do consume more of a standard American diet. So we know, and I know most of you listening are well aware that the gut is probably central to most aspects of human health, and that inflammation, the gut being very much of a host to our immune system that is 24/7 interacting with an environment that ultimately reflects the quality of the outside world, is a central place where either inflammation is being promoted on a chronic basis, or a place where one can significantly modulate or reduce inflammation through healthy lifestyle. And we know that the microbiome is central to these inflammatory pathways and mediators. And whether you're looking at issues of weight and metabolism, whether you're looking at inflammatory issues like asthma, allergies, eczema, and I know many of you are well aware, particularly if you're working in the pediatric population, of how nasty these atopic manifestations can be, many of which are probably being driven at the level of the gut. Autoimmunity in its many forms, from Hashimoto's to rheumatoid arthritis, and the list continues to grow, are being demonstrated or associated with lack of diversity of the human microbiome, overexpression of certain species like Prevotella or Klebsiella. The particular organisms, I think, are not that important because I think our understanding is really still in its infancy. But the point is that proportion is lost and diversification is lost. And for anyone who might have a genetic predisposition for autoimmunity, there's a greater likelihood of ultimate expression. And the same can be said for arthritic issues, metabolic bone disease, other skin issues like eczema, rosacea, a lot of interesting research there linking that to dysbiosis in the gut, to small intestinal bacterial overgrowth, the migration of organisms from the colon into the small intestine. And again, I still think we're at the tip of the iceberg of our understanding of how these mechanisms are related. But even without clarity of understanding, I think there are many opportunities to begin to manage this more effectively through lifestyle, which is the purpose of this webinar. In the behavioral health world, and this, again, I think is fascinating. I do a lot of work with our behavioral health colleagues. Most of our programs are tightly integrated. And as you all well know, you can be working with someone who's a diabetic or maybe has cardiovascular disease. It's unusual these days not to see coexisting depression or anxiety or maybe PTSD or intellectual disabilities, people on the autism spectrum, this fascinating gut-brain connection. And again, I think this is just amazing research that is suggesting important roles between the quality and diversity of one's microbiome and ultimately how their brain works, how they think, how they feel, and this can be extended across the age continuum from young children on the spectrum of autism to older adults confronting Alzheimer's and cognitive decline. Cancer, you can take any chronic complex disease, and these mechanisms continue to draw a very, very strong connection. There's a lot in the literature, as you know. I think if anyone is looking for a nice read and some nice papers, I would highly recommend Erica and Justin Sonnenberg. They are both at Stanford. They have a really good book called The Good Gut, and their research is fantastic. This is a very nice paper from Cell Metabolism. And if you begin to look at why it is that the microbiome of the modern human in 2017 is so different than how it may have been generations ago. And as you know, the explosion of chronic complex disease that we have seen has really emerged over the last few generations, much faster than what genetic mutations can explain. These are alterations of environment that are now so profoundly mismatched with our Book of Life's primal imperative that one is seeing this rapid acceleration. And so this is from the Sonnenberg's article in 2014 from Cell Metabolism. And they make the case that if we, in traditional populations like the Burkina Faso, you see a high consumption of these microbiota accessible carbohydrates. This is essentially fermentable fiber that survives the journey into the colon, where it can then serve as a fermentable fuel for the microbiome. And in those traditional populations, the byproduct of that is more diversity a higher metabolic output, and again, general protection from Western disease. So in the top part of this graphic, the colon and small intestine, obviously there's a world of difference between a broccoli spear and a piece of cake. And even if all things were controlled to be isocaloric, same amount of, for example, carbohydrate, here you would see how the quality of the carbohydrate, in this example, a mac, very much has a different translation. In the example of the cruciferous, most of that carbohydrate is not simple. It will not be broken down quickly, and it will present to this dynamic microbial community an array of fermentable fiber that can set the stage for more effective fermentation and metabolic output, as compared to the standard American diet, where we're just so woefully depleted in the amount of the microbiota accessible carbohydrate that the inevitable byproduct of that is loss of diversity, loss of metabolic output, and a much higher increase in the root causes of chronic complex disease, such as inflammation, such as the hypothalamic pituitary stress response, such as mitochondrial disruption, et cetera, et cetera. And there are many other aspects of our evolving epidemiology, particularly over the last few generations. As we move toward more mono-commodity crops, we have much less diversity in the plants that we consume that have become heavily industrialized. Sanitation, obviously incredibly important from a public health perspective, but the hygiene hypothesis suggesting that our obsession with being germ-free has had the unintentional consequence of interfering with our innate ability as a species to be sharing microbes that might confer more protection throughout life. And then, of course, antibiotics are, as you all know, are in wide use. We prescribe a lot in the United States, and I think although there's much more awareness for the pediatric community as well as the primary care community, many consumers of American healthcare still perceive the antibiotic to be the best way to get from A to B. So I still think there are many educational gaps with respect to how we examine the risk-benefit of what might otherwise seem like an innocuous, relatively low-risk intervention. Probably not at all the case. And we know that almost 1 out of 3 births in the U.S. is from cesarean section. Many kids are bottle-fed instead of breastfed. And I'm not making a judgment statement. There are many reasons for those choices, some of which can be life-altering. We do a lot of C-sections in the U.S., more so than any other country on the planet. And we know that kids who come into the world C-section really, and not surprisingly, their microbial colonization is mostly skin flora. And we know that C-section kids are at a bit more risk for asthma and atopic diseases, eczema. Maybe it's at greater risk for cognitive and learning disabilities. So again, we can begin to see the importance across the life continuum, from prenatal care to how a child is brought into the world, to some of those early years of early childhood development that are so fundamentally important in setting the stage for this microbial community. And so, from a population health perspective, which is where I spend a lot of time in my professional work, what happens at that critical time, that epigenetic period prior to conception, those early childhood years, and then of course throughout life, can really set the stage for how healthy an individual and ultimately the community becomes. And I think, again, we have a lot to learn about this, but it's just a huge public health consideration. So I've touched on some of these noteworthy contributors to why it is we've lost this legacy of microbial diversity. And again, I would say that from a food perspective, it really is a multifactorial, but there's no question most of our food has been stripped of the nutrient density and the fiber that we would otherwise be benefiting from. And if you look at the extent to which, and I know you're all painfully aware of this, refined processed foods, particularly what I would consider carbohydrate dense foods, these tend to be more refined grain-based flour foods. And I'm not suggesting all grains are bad. Whole clearly is better than refined. But there is some, and I'll share some research with you from Ian Spreadbury, who's now at McGill, but carbohydrate dense foods, things that are broken down quickly into sugar, seem to be associated with changes in the microbiome, upregulation of pro-inflammatory pathways, surges of insulin, and so this is, I think, a major nutritional fault line with respect to human health. Again, we've talked about the extent to which the average modern human consumes a fraction of the diversity, both quality and quantity of fermentable fiber that our ancient ancestors once did. We talked about antibiotic use, and it's important to keep in mind that antibiotics, and most antibiotics used in the U.S. are used as part of our agricultural methodology. So we know that many of our industrialized foods, from feedlot cattle to poultry, the milk that cattle produces, the beef that comes from that cattle, the eggs that those chickens produce will have traces of antibiotics in them. So as the saying goes, what we eat is important, but just as important is what we eat ultimately ate. And so we can get microbiotic residues from many industrial foods, and I think that's important from the perspective of understanding that the microbes in your gut will be exquisitely sensitive to even small amounts of an antibiotic. And while it's hard to measure the effect that may be having for any particular individual, it definitely would give one pause, particularly if they're dealing with active inflammatory issues, autoimmunity, quality of life issues, as they consider the foods that they eat, whether they may have antibiotic residues, and to what extent could that be disrupting their health trajectory. I know that this is also a very, I think, open area of research, and while there is still a great deal of uncertainty, if you look at a glyphosate as part of genetically modified foods, Roundup Ready, we know that glyphosate in vitro has significant antimicrobial activity, and we know that even picogram amounts can possibly disrupt not just microbial diversity, but can also disrupt metabolic pathways of the microbial community. One that has been widely studied is the shikimate pathway, which glyphosate will disrupt, that interferes with the microbial community's ability to produce aromatic amino acids like tryptophan, for example, a precursor to serotonin and melatonin and dopamine. And so while the jury may still be out on the risks or benefits of genetically modified foods, it gives me great pause to consider the fact that anything that may have a glyphosate residue that is getting into my system or the system of someone I'm working with could be problematic. And we know that glyphosate now is being found ubiquitously in foods that, like grains, where Roundup is used as a browning methodology to enhance maturity and to bring these crops to harvest. And even organic grain sources are now being shown to have glyphosate residues in them. And so we have a lot more to learn, but I'm reminded every day, as we all are, that, we're all the canaries in the coal mine. And I think as individuals and as caregivers, we simply need to be mindful of potential cause and effect and choose wisely. And as you all know, it can take many years for these things to sometimes reach a critical mass to work their way into mainstream thinking. The hygiene hypothesis, as we've talked about, I think is an interesting and important one. And so as we think about how much time we spend outdoors, how much time our kids spend playing in the dirt, getting dirty, hanging around with pets, and so clearly there is an evolutionary biologic survival advantage of communities of organisms being able to hand off and share microbial communities that, yes, certainly on occasion can be pathogenic, but may have important modulation of immune function and attributes of health over time. Many other aspects of gastrointestinal health and dysbiosis continue to evolve. Many people who've had prior GI infections often never quite regain the microbiome they may have had before that gastroenteritis hit, or they may have noticed changes in how they felt subsequent to that event. We know that people who are getting broad-spectrum antibiotics after hospitalizations, or as they prepare for a surgery, or perhaps being treated for an infection like a community-acquired pneumonia, people will often notice that since those events, they've had things emerge in their health that otherwise have not been easily explained or understood. SIBO, small intestinal bacterial overgrowth, I think is probably very prevalent and very common. It appears to be highly associated and perhaps causal with irritable bowel syndrome. It's clearly associated with enhanced intestinal permeability, rosacea, eczema, and if someone is looking for more of a deep dive there, I would highly recommend Dr. Mark Pimental's work. He is at Cedars-Sinai in California. But again, I think these are all important areas of opportunity for both diagnostic and treatment for anyone who is struggling with a chronic complex health issue that may have some origins in the gut and with dysbiosis. And then many of the medications that I and so many of my colleagues have prescribed through the years and that people can often get over the counter, like these proton pump inhibitors, certainly steroids. We use a lot of corticosteroids in our treatment, and chemotherapy. That while these may be indicated and they may be necessary, I think often they have short windows from a therapeutic toxicity ratio perspective. And we know that PPIs can alter and they increase the risk of small intestinal bacterial overgrowth. They can alter the way that our food is digested and ultimately the extent to which fermentable fiber is able to present itself. So that, again, I think the microbiome just becomes another very nuanced domain for which we think about all aspects of health and how treatments, which otherwise may have an indication, often go on for long periods of time and can become disruptive. This is one of many reviews that has looked at the differences in the microbiota of obese versus lean. And on the left, we know that the microbiota community does tend to be much less diverse. Many people have found, though it's somewhat controversial, that Firmicutes species are often more predominant. But you see alterations in these mechanisms that alter satiety and increase lipopolysaccharide, inflammation, fatty liver, alterations of these signaling molecules like GLP-1, AMPK. These are all metabolic acupressure points, if you will, that are modulated in ways that promote fat storage, that upregulate inflammation, that diminish the extent to which we can burn fat as a fuel, etc. You see just the opposite in lean microbiota on the right. Much more diversity, more Bacteroidetes, and you see very different immunologic, gut hormonal, and fat oxidation expression in those individuals. And again, there are just a number of papers now that are connecting the gut microbiota with energy, inflammation, and while I think the jury is still out on what this means from a therapeutic potential, it is clear that lifestyle can influence one's microbiota, and probably in a matter of days to weeks, you can begin to demonstrate shifts based on changes in diet, recognition of what might be SIBO and reconciliation of that, etc. We know, again, interesting research that if you take the microbiota from a woman who was pregnant in her 3rd trimester, and under those circumstances, physiologically, women need to be extracting more calories from the food that they ingest to promote the growth and development of the fetus. If you take the altered microbiota, in this case altered in a positive, physiologically important way, and you transplant them into a lean mouse, you will see that mouse gain weight and become more insulin resistant. So this is an example of insulin resistance as an important adaptive mechanism, probably in part facilitated by changes of the gut microbial community that can influence human health. And many women, will share with me after they've had kids, that they've never really gotten back to their pre-pregnancy weight. And sometimes I do think changes in the microbiota may be in part responsible for that never returned to the prior baseline. And again, endotoxemia, this concept of dysbiosis and LPS overexpression and permeability altering systemic inflammatory responses is clearly linked to obesity, cardiovascular risk, diabetes, insulin resistance, etc. And these are comorbidities that are frequently clustered. Rarely do I see someone who's insulin resistant who isn't also struggling with blood pressure or dyslipidemia or weight or depression. And it's not because, in my opinion, they've drawn an unhealthy hand of cards. They have environmental mismatches with their genetic predispositions that is setting a metabolic landscape that is a common root cause of all of those different phenotypes, if you will. So again, in this graphic, we know that LPS can permeate the layers of the intestinal lining in response to the standard American diet. And people who are obese and diabetic and have fatty liver have much more LPS, have much more inflammatory upregulation as measured by C-reactive protein. And you see these cycles that continue to perpetuate disease progression. This is some work by Ian Spreadbury that I alluded to earlier. And basically, Dr. Spreadbury makes the point that one of the big changes in modern diets is that we do consume more of these acellular carbohydrate-dense foods. These tend to be grain-based foods where the preponderance of that food is carbohydrate by weight, and they tend to be broken down quickly and turn into sugars very quickly. These are much more simple carbohydrates. And if you look at the black bars up top compared to the white bars, the black bars represent foods that are very commonly consumed in the modern diet. The white, more common in ancestral diets. And on the x-axis below is carbohydrate density. Most ancestral diets are rare to consume foods that have a carbohydrate density above, say, 25 grams per 100 gram of that food, as compared with the standard American diet, which is so grain-based, refined grain-based, that again has a very high carbohydrate density. And carbohydrate density and glycemic index don't always go hand in hand. So, things like bread, things like oatmeal, there are some carbohydrate-dense foods that may have less of a glycemic effect but still may create significant problems for one's gut microbiota. And what this busy slide demonstrates is if on the upper left, if you look at fiber that's coming from living cellular sources like vegetables, fruits, beans, legumes, you get a very different effect inside the small intestine. These do not break down quickly. They tend to have limited effect in the small intestine. They're able to present to the colon where they serve as a fermentable fiber, and generally you have more adaptive and resilient metabolic systems. If you contrast that with diets that are very high in these carbohydrate-dense foods, you see just the opposite. And it starts in the mouth with what we know are very high prevalences of periodontal disease, where the microbiota of the oral cavity will quickly break down these more simple starches, produce a pro-inflammatory response, and the dentist may call that a cavity, they may call it an abscess that needs a root canal, they may call it periodontal disease. The bottom line is this is probably a potent adverse byproduct of a mismatch between microbial community and the food that they are fermenting. And then as you go downstream, you can appreciate that the upregulation of inflammation in the microbial community of the gut will create systemic inflammation, which we know puts people at risk for cardiovascular disease, cancer, obesity, diabetes, etc. I just wanted to add a slide on non-nutritive sweeteners. This is a comment that comes up often. There's very little human data, but if you look at what's been published in animal studies, if you give a non-nutritive sweetener, in this study, this was published in Nature, widely disseminated, I believe they used sucralose in this study. There were significant changes seen in the gut microbiome of individuals taking non-nutritive sweeteners that were associated with higher insulin levels and dysglycemia or glucose intolerance. So just by way of a summary, this is a little bit geeky from a metabolic perspective, but again, we know that many of these metabolic pathways are influenced by the quality and diversity of the microbes, and so these are fault lines, all of which in some way can be addressed by healthier lifestyle. And so in the last 5 or 10 minutes, let's just look at, we've already touched on a lot of how lifestyle impacts this. Certainly removing foods that can be problematic, and that may take the form of an elimination diet, particularly with an emphasis on sugars, fructose, and these refined carbohydrate-dense foods. We know that people, particularly people who've been on proton pump inhibitors for a long time, might benefit from digestive enzymes to help more effectively break down foods for microbial fermentation and assimilation. We know that pre and probiotics, though human studies are still somewhat limited, have a really important role. Supplements may have a role for some. I don't want to get too into that. And of course, lifestyle and certainly all of the things that we know to be good for us, from movement to mindfulness to social connection, probably do via pathways of the central nervous system and brain-gut enteric system can be significantly beneficial. So the main issue is really getting more of these microbiota accessible carbohydrates from soluble fibers, as you see listed as some examples, to the non-starch polysaccharides, these from onions, garlic, Jerusalem artichoke, leeks. These are really wonderful forms of fermentable fiber, to more resistant starch, that you'll find in beans, legumes, lentils, cooked and cooled potatoes, plantains. These can be really helpful and I think most important in beginning to redevelop the diversity and balance of one's microbiome. And again, there are many prebiotic-rich foods. A few of these slides I got from Kathy Swift. I want to thank her for that. And I know you're well aware of the importance of these foods, as they are integral to all that we are talking about here. And again, this can take some time and some education in terms of helping people understand the best sources of these fermentable fiber foods, as well as preparation. But they make a huge difference. And we know that many herbs and spices have significant antimicrobial activity in instances of overgrowth and anti-inflammatory properties. So, I use a lot of ginger, peppermint, turmeric, black pepper. These are just such an important part of that whole foods approach to the gut and to health in general. And we talked a little bit about butyrate, a short-chain fatty acid, and we know that some research has shown that giving people probiotics, and certainly we saw in the Burkina Faso that one can upregulate GLP with more fermentable fiber, and that's very important in insulin sensitivity, insulin signaling, sugar management. I mean, there are many expensive prescriptions that we give that have GLP-1 effects. One can reproduce those effects through thoughtful interactions between food and the gut. And again, these are just examples of studies that show that once you start to increase GLP-1, your metabolism goes up, you start using more fat as a fuel, and that appears to be the case throughout the age continuum. And there are many studies that have looked at probiotics. I think probiotics can be helpful in modulating inflammation. Most of the research is limited with respect to whether or not probiotics can actually change in a more enduring way the diversity of one's microbiome. I do think more fermentable fiber and prebiotics is a better strategy to achieve that. But this is an example of a randomized placebo-controlled trial showing probiotics helping with insulin sensitivity and fat oxidation and weight loss. There are several trials that have been published. Again, these tend to be small trials, but again, this is just looking at certain strains of lactobacillus that seem to mediate higher metabolism. A more recent meta-analysis looked at 17 randomized controlled trials, and many did show positive effects, but not all of these organisms are the same. In this review, if you look at Lactobacillus plantarum and Gasseri, more effect than what you saw with Acidophilus. So again, I don't think one can draw specific conclusions other than to say for some, particularly if they have lots of inflammation, asthma, eczema, rheumatologic complaints, pain, fatigue. I do think probiotics, fermented foods, which I should have mentioned, like yogurt and kimchi and sauerkraut, miso, tempeh, play very, very important roles from a more food as medicine perspective. And this is just another, this is looking at fermented milk on adipose and metabolism. Many studies have also shown fermented milk products can enhance mood, and you actually see changes on functional MRI of the brain that are associated with these foods. So just a fascinating area that continues to evolve. So by way of summary, the most, I think, robust lifestyle changes to bring diversity and balance to the gut microbiome would include diminishing sugar and these processed, refined, carbohydrate-dense foods. One might consider an elimination short-term of grains and gluten. And you're all aware of the fact that some may respond, others not at all. Glyphosate exposure, glyphosate gives me pause. And, these are huge topics. And there are a lot of people that think that glyphosate is totally harmless. There are so many lines of evidence, in my view, that would suggest the opposite of that. So it is something I think about all the time and introduce into the partnerships that I form with patients and my colleagues. We talked about fermentable fiber and reviewed some sources of that. glyphosate as well. Being careful with feedlot meats, poultry, eggs as subtle sources of antibiotics. Non-nutritive sweeteners, even emulsifiers now, I think are generating some pause with respect to the microbiome and gut permeability. Again, I think we have a lot more to learn about this, but it just makes sense if someone is able to consider these in their lives, and not everyone may be able to keep it as clean as possible. Testing, I think, can be important, and that's a big topic. I'm always meeting people where they're at. A lot of people I work with don't have a lot of resources, so I'm very careful with out-of-pocket cost. But SIBO and testing for SIBO with breath testing is usually covered by insurance, can be very helpful. I think stool testing is probably one of the more meaningful and actionable tests of the gut microbiome that I order most commonly. We talked about probiotics. There is no one right probiotic. How much and how diverse, I think, are sort of open issues. But generally, I'm trying to get 10 to 20 billion colony-forming units a day. If somebody's really got some angry issues, I may aim higher for that with something like VSL number 3. And again, I think this whole area of fecal microbiota transplants is an interesting one, and as many of you know, for C. difficile, which is a horrible hospital-acquired infection, it will remit most people, and most of the trials have looked at the most resistant of cases, and just another example of how, in a very short time, altering the population of that ecosystem can bring profound traction to health and one's life. So that was a bit of a whirlwind tour there, and I apologize that I dropped in a lot of content there in a short time. It's a topic that has so much to it, and I do have time. Let's see, I've got some questions here. And I think I've got time for one question. So this question, this is from James, and thank you, James. Can you comment on any research you have come across regarding pesticide and herbicide residues, primarily glyphosate, found in common foods, and how it affects the microbiota? So I think there is some growing research there. Anthony Samsel and Stephanie Seneff are both doing, I think Anthony Samsel's name is S-A-M-S-E-L. Stephanie Seneff, S-E-N-E-F-F, she is at MIT. Clearly they are showing significant antimicrobial activities of glyphosate. Research looking at the shikmate pathway and alterations of aromatic amino acid use is research that continues to grow. Glyphosate as a chelator, and if you look at the extent to which glyphosate has the potential to chelate important nutrients, I think is really important when you look at the quality of the soil. And so I think this is an area that is going to continue to grow dramatically in terms of our insights and understandings. But Stephanie Seneff and Anthony Samsel are two researchers I would guide you to. And I'm really sorry that's the only question that I have time for. But my hope is that maybe I can work with the folks at Functional Formularies to try to address some of these offline. And I do want to remind everyone that this is being recorded and will be on the Functional Formulary website for future use, and I believe my slides will be available as well. And CPE certification for the dietitians will be emailed over the next 48 hours, so I just wanted to remind you about that. And I know that you all have busy days to return to, so most grateful for your checking in, and keep serving those you serve as you do. And Be well and stay well. Thank you.